And Derek, stop spamming chat. I can't read. Can people see my screen share? You can't hear me? Oh. Let me see. Is this better? Can you hear me now? Oh, bruh. Okay, Ivan. Can you guys hear me? Can you see my screen share? Type in chat if you can. Okay. Yeah. Okay. Then we can get started. Um, so is it recording? Yeah, we're rolling. So today we're going to be covering chapters 14, 15, and 16 of Campbell's. Let's start with chapter 14. It concerns the Mendel and the gene idea. This is some vocab. There's really not much to talk about here. A homozygote has two identical alleles, either two dominant or two recessive. Heterozygote is by Derek. Heterozygote is a trait that has an organism that has two alleles, two versions of alleles for a given trait, one recessive, one dominant. Phenotype, genotype, test cross. I'll give you guys like 15 seconds to read over this. Another thing, a gene, every normal person has two copies of each gene, and each copy of one copy of a gene is called an allele. So everyone has two alleles per gene because they have two chromosomes. Genotype, test cross. Well, test crosses we'll go over in a bit. Yeah. So laws of inheritance. A little bit about Mendel. Mendel was the guy that came up with Derek, stop. Mendel was the researcher who came up with the laws of inheritance. He experimented with pea plants and discovered that traits were passed on. He came up with a few rules that we'll go over in a bit. Another definition, a true breeding individual is an individual that with repeated breedings with other true breeding individuals will continue producing one phenotype. So for example, if you have two purple pea plants with purple leaves, then a true breeding phenotype would be a true breeding phenotype would be plants that kept on producing purple plants. Here's some more definitions: P generation, F1 generation, F2 generation. So in a genetic study, the first set of plants that are interbred are called the P generation. Then their offspring is called F1. The offspring of F1 is called F2. Laws of inheritance. So this is one of Mendel's laws, the law of segregation. It basically says that each parent has two alleles. So they have two chromosomes, and each chromosome has one allele per gene. But a parent will only pass down one allele to their offspring. There are two types of alleles in like simplified Mendelian genetics. There's the dominant allele, which will code for a functional protein. It will do some kind of function. And the presence of a dominant allele, no matter if it's one or two, will lead to a change in phenotype. Whereas the recessive allele has no noticeable effect. So it might like code for a non-functional protein, or basically a recessive allele will not have any effect on the phenotype. However, if you have two recessive alleles, the absence of function leads to the phenotype. Does that make sense? So dominant allele, function leads to phenotype. Two recessive, absence of function leads to phenotype. The two alleles for a trait separate during meiosis. So this is called the law of independent assortment. We'll go over this in a bit. But when meiosis happens, the gametes, the resulting gametes only have one chromosome per gamete. And each chromosome will carry one allele. If you're going to, the Punnett squares are essential for Mendelian genetics and the use of it in general. It's probably extremely helpful for the opens. It's this square thing in the corner where you can use the genotypes of the parents to find out the phenotypes and genotypes of the offspring. Inheritance patterns, degrees of dominance. So like I said, like I alluded to before, inheritance is not as simple as complete dominance, where either the presence of a dominant allele will create a function, absence of a dominant allele will lead to a lack of function, which leads to phenotype. There are actually many kinds of inheritance patterns. The first of which is complete dominance. This is the one that we think of usually when we think of Mendelian genetics. This is where if you have an allele, let's take flowers as an example, right? If you have a allele for red flower color that's dominant to an allele for white flower color, if you have a heterozygous individual, meaning that the genotype, they'll have one red allele and one white allele that will lead to a red or dominant phenotype. In this case, the dominant allele completely masks over the recessive allele, which is why it's called complete dominance. Incomplete dominance, on the other hand, is kind of known as the mixing of phenotypes. So if you have a dominant red allele and a dominant white allele, if a plant has a red petal allele and a white petal allele, instead of becoming the petal being red, all the petals will be pink. So it's a good way to think of incomplete dominance is a gradient of different phenotypes, right? Heterozygous offspring look like a mix of the two parents, and codominance. So codominance is different from both complete and incomplete dominance. It's not, one trait does not overshadow the other. One allele does not overshadow the other. What I mean by this is, say you have a one allele for red flower color and another allele for white flower color. If you have a heterozygous plant with a red and white allele, you won't have red flowers or pink flowers. You'll have a combination of red and white flowers. So the two alleles don't mix and they don't like interact with each other. Any questions? Okay. One important example of codominance is in blood types, where you have the A allele and the B allele are codominant, meaning if you have the phenotype IAIB, then you'll have blood type AB. But if both of these alleles are dominant to the allele for O type blood, which is known as little I. Okay. Inheritance patterns, non-Mendelian genetics. So when we went over Mendelian genetics, it was generally with a Punnett square. It's very simple. Each parent contributes one gene per trait. In reality, that's really not the case. Like most things, the real world is actually a lot more complicated. So here are some examples of exceptions to Mendelian genetics. The first of which is pleiotropy. Pleiotropy just means a gene has multiple phenotypic effects. So like maybe genes that code for transcription factors or Hox genes, homeobox genes, the genes that have effects on the entire organism as a whole, they will show pleiotropy, whereas generally Mendelian genes affect one single trait. Epistasis is different from pleiotropy. Of course, it's different from pleiotropy. This is the expression of one gene. This is where the expression of one gene at one locus affects the expression of another gene at a different locus. Now, what does this mean in practice? It basically means that whether or not one gene is expressed will affect a different gene, a completely not unrelated, but different gene. So for example, this is an example in Campbell's. There, the coat color for Labradors is it's determined by two sets of genes. The first is determining the coat color, right? This is like normal Mendelian genetics. So you have the dominant allele B for like black coat color, recessive allele little b for brown coat color. But there's a different allele, there's a different gene that will affect coat color as a whole. This gene codes for whether or not pigment is produced. And so you see how if there's no pigment being produced, then there will be no coat color. So all the Labradors in this corner, they're all like yellowish white. They have no pigment in their coats. Does that make sense? So epistasis is where one like control gene, for example, the gene that codes for pigment production, will affect a greater phenotypic trait. For example, in this case, the coat color. All right. And polygenic inheritance is traits vary on a gradient. Like this basically means that a single trait is affected by many different genes. For example, like your skin color, there's no one allele that affects skin color. But a lot of genes, I would even go to say most genes are polygenic, or most traits are polygenic. My apologies. Questions? All right. Oh, what is going on here? Huh? Hold on. Let me see if I can. Right. Oh. I thought the stuff behind was a different slide. This is an example of polygenic inheritance, where you see there are 3 different alleles that affect. So there are 3 different genes. Each of these dots is a gene, and each like specific dot represents an allele. So you can see how different combinations of these of sperm and eggs carrying different combinations of alleles can result in a gradient of different skin tones. Let's just say, like, does that make sense? Okay, more definitions. Multifactorial genes are genes that are affected by not just the allele. So that's just a fancy way of saying like the environment can affect the phenotype. This is also an example from Campbell's. These flowers, hydrangeas, their genotype gives them a certain flower color, but depending on the substrate of soil that they're grown in, they'll absorb different things and they'll develop differently, leading to different flower colors. Here are some human inheritance patterns. There's generally, when we talk about inheritance, you have 2 types of chromosomes, right? Generally speaking, you have your autosomes, which are the 44 chromosomes that determine somatic cell functions, and then you have your sex chromosomes, 2 sex chromosomes, 44 autosomes, right? So a trait can either be autosomal recessive or dominant, which means that autosomal, it's carried on an autosome. Wait, it's carried on an autosomal chromosome. What is going on? It's carried on an autosomal chromosome, and it's either recessive or dominant. If it's autosomal recessive, then it's carried on an autosome and it's a recessive gene. If it's dominant, yeah, you get the idea. An X-linked recessive or dominant gene is oh wait there. An X-linked recessive gene is a gene is a gene that is found only on your X chromosome, specifically like the XX chromosome. That's pretty self-explanatory. Does anyone know why there are very few Y-linked recessive or dominant genes? It's kind of a weird question, but why the Y chromosome is very small. Most of the genes there code for function of the male reproductive system and development of the male reproductive system. So it wouldn't make sense for there to be. It's just very small. There's no space for a gene. Here's an example of a pedigree. So, a little bit about how to read a pedigree. The squares represent males. The circles represent females. If the shape is shaded, like this square is shaded, the circle is shaded, it means that they are affected by the trait. So in this case, it's autosomal recessive. So we know that this guy and this girl both have 2 copies of the autosomal recessive gene. And then the pedigrees show family history, and it shows you how the trait has been inherited, where it originated from. Excuse me. Yeah. Autosomal dominant. Notice how here in this autosomal recessive chart, there were a lot more. So you see how there in the F1, so this is a P generation, F1 generation, F2 generation. That's how we can just define them like that. You can see how here there's a lot more individuals who don't express the phenotype, right? But in the next slide, autosomal dominant, the difference between dominant and recessive genes is that if you have a dominant gene, then you'll automatically express the phenotype. But if you don't, but for recessive phenotypes, you have to have both copies of recessive genes. So in this case, you can see how there are a higher percentage of the offspring have gained the phenotype because it's dominant as opposed to before where it was recessive, right? And this is X-linked. Something about X-linked genes. X-linked recessive genes are actually, I think we'll see it on the next slide. So I'll just talk a little bit about this. Males only have 1 X chromosome, or they have 1 X and 1 Y chromosome. So in X-linked dominant pedigrees and problems, it's easier to find out the phenotype of the male than it is usually of a female. For example, up here, where my mouse, can we all see my mouse? The square label number 2. We know that this is a male. He has 1 X chromosome, which means, and he doesn't express the phenotype, which means that he does not have he is recessive for this gene. He is recessive for this phenotype, right? Because the gene is dominant, and since he doesn't have it, he has a recessive version. Yeah, pay attention to the, yes, I did. I'll just reiterate just in case I didn't. Black means affected, white means they're unaffected. But like they don't express the phenotype. And here's X-linked recessive. So notice how in this graph, there are a lot more afflicted males than there are afflicted females. Anyone have a guess as to why that is? 5, 4, 3, 2, 1. Okay. The reason for this is because males only have I'll just explain it. Uh, yeah, but uh, yeah, I guess that's not a bad answer. The males only have one copy of the X chromosome. They don't have redundancies. So if they get one X recessive, then they automatically are affected by the condition. Whereas females, if they have if they can afford to have one X recessive, if the other because they have another X chromosome that could contain the X dominant, they have a backup. Does that make sense? So because males only have one X chromosome, you should pay attention to them if the problem is talking about X-linked. Yeah. Human recessive disorders. For recessive disorders, there can be carriers, or a carrier is an individual who is like phenotypically. A carrier is an individual who is heterozygous for the recessive disorder, which means they're not affected by it, but they carry one recessive allele. And if they mate with another person who is also a carrier or who does have a recessive allele, they could pass on. Both of the parents could pass on the recessive trait, and their offspring could get the disease. So, some important examples: cystic fibrosis. This is like a buildup of mucus. You lack in one specific transporter. Albinism, you lack an enzyme that makes pigments, melanin. Sickle cell disease, this is a big one. If you're homozygous for sickle cell disease, all these are recessive, by the way. If you're homozygous for sickle cell disease, you sorry. If you're homozygous for the sickle cell trait, if you're homozygous for the trait, then you get sickle cell disease. That means that your blood cells spindle very easily. They form like scythe-shaped things. They clog up your capillaries. That's generally bad. However, the heterozygote will only experience mild blood spindling, and as a result, that will give them some immunity to some resistance to malaria. And that's why sickle cell this trait, although the homozygous is bad, the heterozygous is actually good. So that's why it's persisted. Dominant disorders, anyone who has a copy of the dominant trait will be affected. And as a result, these alleles are much more rare within the population. One example, Huntington's disease, that's a big one. It's caused by a protein that has like a lot of short like repeats in its exons, which makes an error in how the protein is like made and stuff. Yeah, you can test for dominant alleles or just in general genetic disorders through one of two ways: amniocentesis or CVS, chorionic villus sampling. So amniocentesis is when you take like a needle guided by ultrasound. You extract like 10 milliliters of amniotic fluid, and then find an embryonic cell from there, culture it, and determine the karyotype. You can also sequence their DNA to find out if they have genetic diseases. Karyotypes are usually just for like seeing aneuploidies, or we'll get into that in a bit. Chorionic villus sampling is the same thing, except you're getting the tissue from a piece of the placenta, not the amniotic sac. These are considered invasive procedures because they generally involve like puncture and extraction. Before you consider doing like an invasive procedure, usually you have like screenings, like the ultrasound thing where you see a gray blurry image of the fetus, not infant. That's a type of screening where you can that shows you risk factors for whether or not you should do a invasive procedure. And we're done with 14. Any questions on 14? All right, I'll get right into 15. So there are many types. You might think that all animals have sex determined by an X and a Y chromosome. That's not really the case. There are many, like on this right side, there are many different types um of sex determination in different animals. Humans have the XY system, like I just said, where the male has a Y chromosome, and that Y chromosome will determine the sex of the offspring. Um, there are other systems where there's the XO system, which basically means that if you have an even number of X chromosomes, then you're a female. If you have an odd number of X chromosomes, you're a male. There's a ZW system where instead of the male carrying the Y chromosome and determining the sex of the offspring, the female carries a W allele, and the presence of the W allele makes the animal a female. Yeah, there's not much to say. And then there's the haplodiploid system where for example, like bees, if you have a diploid genome, which means you have 2 copies of every chromosome, then you're a female. If you're haploid and you have 1 copy of each chromosome, then you're a male. Yeah, develop unfertilized eggs. Females develop from fertilized eggs. Make sense? All right. Mothers pass on X chromosomes, father pass on either X or Y. Each sex chromosome assorted independently. This is the law I was mentioning earlier. I think there's a slide on it later. The law of independent assortment, which states that you have all of your somatic cells and your germ cells have 2 sets of alleles. How those alleles are split into how those chromosomes are split into different gametes is completely independent of each other. So yeah, I mean, I don't really know how better to explain that. If anyone has any questions, DM me. For example, an example of an X recessive disorder is color blindness. All colorblind daughters have colorblind fathers and at least carrier mothers. This is because color blindness is X recessive. Daughters have 2 X chromosomes. So, to be colorblind, the daughter needs 2 copies of the recessive allele. That means that the father must carry 1 and the mother must carry 1. If the father carries a recessive allele because he only has 1 X chromosome, then he must also be colorblind. But the mother can she can carry a dominant and a recessive allele, but and still pass on the recessive allele, which could still result in a colorblind daughter. Yeah. Here are some sex-linked genes. Colorblindness, X recessive. Hemophilia, I think, is also X recessive. Unless I'm throwing. Duchenne muscular dystrophy. I think these are all. I hope I'm not throwing. Yeah. Sex-linked genes, X inactivation in females. Oh, this is really cool. Yay! Thank you, Derek. Being useful for once, I see. X-linked genes do make proteins. What do you mean? Oh, X inactivation. The reason for X inactivation is to stop excess protein production. As we'll see later in like Klinefelter syndrome, if you have 2 X chromosomes, you get double the amount of proteins from the X chromosome, and that's quite bad. Actually, no, sorry, not quite bad. It's bad, but like not devastating. When I learned about deactivation of bar bodies, deactivation. Sorry, let me restart. So a female, sorry, a female has 2 copies of an X chromosome, right? but they only want to express one copy of it. So they go right there. So they'll deactivate one copy and condense it into a small structure called a bar body. How do they know which chromosome to condense? This is actually decided really early embryonically. When the fetus is still in its like blastula gastrula stage, one X chromosome is inactivated by random, but it's not. The X chromosome can be inactivated. A different X chromosome could be inactivated in every one of the cells. One cell could inactivate the father's X chromosome, the other can inactivate the mother's. As a result, when these cells divide and form the fetus, some cells will have the X chromosome active. The active X chromosome will be from the father. Some cells will have an active X chromosome from the mother. Also, this is because the pattern of inactivation is preserved. This one example that you might need to know is tortoiseshell cats. The cats with like patchy fur, like orange, brown, orange, gray, white patches. That is because those patches of fur have different inactivated X chromosomes. Patches of skins lacking sweat glands in humans. This is another type of disease. I don't think you need to know this. Let's see. Genes located near each other tend to be inherited together. Oh, linked genes. So, this has to do with the concept of crossing over. So generally, do we all know what crossing over is? Crossing over is during the first phase of meiosis, where you have two homologous tetrads, which is just to say 4 copies of each chromosome, and the DNA forms like something called a holiday junction, and then it exchanges DNA based on homologous sections. You can exchange alleles through that. In this case, this is talking about how if 2 genes are very close together, then those genes are, if they're crossed over, they're most likely going to be crossed over together. If you think of it like, if there's a lot of space between two genes, then there's a lot of space to split the genes in half when you do crossing over. So if the genes are close together, they're usually going to be inherited together, and these are called linked genes. Does that make sense? I feel like I could have explained that better. Carrying on. Linked genes. No recombination occurs. Remember when we had I don't think we, oh no, we do. Let me just go back. Right here in this corner. This is a Mendelian tree for 2 different genes, right? So you see how there's one allele for coat color and one allele for presence of pigment. And you can see how if the genes are unlinked, then they can result in 16 different phenotypes, right? But let's go back to where we were. Here, if the genes are linked, then there are only so many combinations of genes in the gametes that you can get. And that results in fewer phenotypes. In this case, the dominant genes are inherited together, the recessive version of the genes are inherited together, and that results in all of this. Yeah. Questions? Percent recombination, okay, so even if genes are close together and they're linked, they sometimes, like, by random chance, those genes could be separated just because the holiday junction just happened to cut right between the two genes. And so that will result right here. See, normally the alleles are linked to dominant and recessive, but a recombinant offspring is just, it's a okay. You do you see how the alleles for that made up the recombinant offspring have one dominant, one recessive, one recessive, one dominant? It just means that when the alleles are supposed to be linked, a recombinant offspring is an offspring that the alleles are switched up and they're no longer linked. Crossing over happens, and yeah. Does that make sense? Get back to me if it doesn't make sense. Yeah. So we can calculate how many map units away, how far apart two genes are by how many recombinant offspring there are. So let me try to explain this better. Basically, it's what percent of the offspring are heterozygous for one, are made by a heterozygous gamete. So the percent recombination allows you to determine how, like, relatively how far away the genes are. Yeah. 15.4, alteration of chromosome number. Abnormal chromosome numbers could be caused by non-disjunction. Homologous chromosomes or sister chromatids do not separate properly. Incorrect number of chromosomes is aneuploidy. Yeah. So in meiosis, when you have like the two tetrads lining up, the four copies of a chromosome lining up, sometimes they don't separate properly, right? Maybe all four copies will go to one side of the cell during the anaphase, or during meiosis 2, when there's a pair of chromosomes lined up on the middle, maybe both chromosomes will go to one side. And as a result, one gamete will have two copies of a chromosome. The other gamete will have zero copies of that chromosome. This will result in aneuploidy. That's just a definition of having an incorrect number of chromosomes. If you're lacking one chromosome, you're monosomic. If you have an extra chromosome, you're trisomic. This is very problematic because, unlike X chromosomes, there's no innate mechanism to disable a gene. So you get double the protein product, and that really messes up like the intricacy of biological systems. This aneuploidy is not to be confused with polyploidy. Aneuploidy means you have a duplication or an absence of a single allele, whereas polyploidy is a duplication of the entire the genome. So let's say an example of polyploidy in humans would be like if you have a human with 92 chromosomes. That's not aneuploidy because you're doubling every single chromosome that you have, resulting in two sets. This is, in animals, this is almost always deadly. Like usually aneuploidy is deadly by itself, so polyploidy is much worse. But in plants are sedentary. They don't have as complicated a system. So they can live with polyploidy. And that's why strawberry plants are like octoploid. I think corn is hexaploid or something. They have many copies of their genome. And this is important because this allows for speciation. So if you have multiple copies of a genome, one copy of that genome can be making normal proteins as such. The other copy can deviate and mutate to make proteins with novel functions. And those novel functions can help you make a new species. Alterations of chromosome structure: deletion, duplication, inversion, translocation. Deletion, chromosome fragments lost; duplication, double number of a gene; inversion, sometimes if you try to cut out a piece of gene, it'll like, actually no, not cut out, but the gene might just be attached in the reverse order. And translocation, a gene is moved to another section of a chromosome. This happens when you have homologous regions between two non-homologous tetrads, and they switch pieces of DNA even though they're not supposed to. The terms are pretty self-explanatory. It's in the name. Here are some notable disorders due to chromosome alteration. Trisomy 21 is Down syndrome. You have 3 chromosome 21. One general trend you can see with chromosomal alteration is that the lower the chromosome number is, the more deadly the aneuploidy is. So 21 is, that's because the lower the number, the bigger the chromosome, the larger the number of genes, and therefore the higher the effect a duplication has on the phenotype. Trisomy 21 is an extra chromosome 21. 21 is like on the smaller side, and as a result, people with Down syndrome, they have like different facial features. They can be like slightly mentally retarded, but many of them can be like productive members of society. Most of them don't have very serious disability. What's up? That's not a slur, that's a diagnosis. Aneuploidy of sex chromosomes. So this is what I was talking about before, where if you have an extra X chromosome, then that could be bad. So in Klinefelters, it's a biological male who has an extra copy of the X chromosome. This generally results in a taller stature, slimmer. You're taller, you're more slim, and you have more feminine features because of your X chromosome. I think sometimes your testes don't develop properly. Yeah, I've never heard of an extra Y chromosome. I don't think the extra Y chromosome will be that harmful or effective in any way because it's so small, right? Trisomy X, in females, if you have 3 X chromosomes, I don't think this is that big of it. I'm not sure. Actually, I've never heard of trisomy X or extra Y. I don't think trisomy X is that big of a deal either, because you can just inactivate both. That would make sense. I did read Cannibals. I don't think this was in there. It probably was. I probably didn't. And Turner syndrome, you have it's a biological female missing an X chromosome. Generally, this leads to an underdeveloped female reproductive system, and some people who are with Turner syndrome are sterile. Altered chromosomes, pre-duce shot. This is a infantilely fatal. It's a fatal deletion in chromosome 5, which is called pre-duce shot because the observed people with the disease, the physicians describe their call as like the crying of a cat. Another famous one is the Philadelphia chromosome. You take like a piece of chromosome 9, a tiny piece of chromosome 9, and exchange it with a piece of chromosome 22, and that results in chronic myelogenous leukemia. So because of the way that promoters are arranged, the exchange of DNA makes the proliferation of blood cells, the gene for that, more readily accessible. CML. Exceptions to inheritance. Genomic imprinting. This is actually quite cool. So you can the way you live your life will affect how your the way you live your life will affect your offspring, I think, because your how your life will change how your DNA is organized, and the organization of DNA, like methylation. When we say methylation, it's usually of DNA pairs, DNA bases themselves, usually cytosine. When you how you live your life will determine how your DNA is imprinted, how it's edited, how it's made, and then that will be passed on to your offspring. Organelle genes, extranuclear genes. One big example, mitochondrial genes are only passed on from the mother, and this can lead to some like ragged red fiber. That's a mitochondrial disease. Yeah. And we're on the Campbell 16. Any questions? I might have to speed through this one. All right. DNA is genetic material. Hershey and Chase, it's a pretty famous experiment where they inserted radioactive bits of like material into the either phosphorus or sulfur into a bacterial cell, and if DNA was being replicated, then the radioactive phosphorus would be present in like future generations. If proteins were were the hereditary material, then the radioactive sulfur would be present in future generations. And they found that DNA, because radioactive phosphorus was found in offspring, DNA is the hereditary material. Bacteria can undergo transformation. Basically, bacteria can uptake extra, like if there's DNA floating around in substrate, bacteria can like swim over to it and take it up, bring it into their, incorporate into their genome. This is like plasmids. They can take up plasmids. Specifically, the one of importance is R plasmids. They can obtain resistance from genes in substrate. Another example is what should I call it? The one where prophages. So sometimes pieces of DNA from bacteriophages can lead to a virulent phenotype of the bacteria, and if a bacteria takes up virulent dangerous DNA, then it will itself transform into a virulent DNA. Chargaff's rule. This is something that you should just like know based on intuition. It says the amount of adenine is equal to the amount of thymine. The amount of cytosine is equal to the amount of guanine. This is just because these two are base pairing. So every single time you have an adenine, it will be paired with a thymine in the DNA. Yeah, structure of DNA. Rosalind Franklin's photo 51. Watson and Crick basically didn't credit Franklin, but she was a big discoverer in DNA. She supplied, like, her work supplied the photo for this, which allowed Watson and Crick to to deduce that DNA is antiparallel. It's made up, the backbone of DNA is phosphate and a 5-carbon sugar in a repeating chain, and attached to the sugar are nitrogenous bases. So the structure of a nucleotide, which is a monomer for DNA, consists of a phosphate, sugar, which is deoxyribose, and a nucleotide base, the nitrogenous base. I'm sorry. Another thing that you should know, purines versus pyrimidines. So purines are the big rings. So if you see DNA, sometimes you'll see the individual nucleotides portrayed as either 2 big rings or 1 small ring. The 2 big rings are called purines. The 1 small ring is called a pyrimidine. Make sense? All right. DNA replication follows a semi-conservative model. Parent strand split, new daughter strand. So this is just saying that when DNA is replicated, the parent strands are used as a template and are and one strand, each new strand of DNA consists of one old strand and one new strand synthesized newly from free nucleotides or nucleosides, whatever. These are the steps of DNA replication. It begins at origins of replications. This is true for both bacteria and eukaryotes, except in eukaryotes, as you can see, there are many origins of replication, right? In bacteria, there's only one. Usually there's only one. At replication forks, the helicase, so here, the helicase will first open up the DNA. The topoisomerase will be ahead of helicase. It will occasionally like cut out pieces of DNA and put it back in a way that prevents supercoiling. It preserves the integrity of the DNA. And single strand binding proteins will bind to nucleotides, and nucleotides are originally held together by hydrogen bonds. And sometimes if there weren't single stranded binding proteins, after helicase comes through, they'll just hydrogen bond back together. But because there are single stranded binding proteins, it prevents the DNA from like sticking together. Okay, bye, Derek. Yeah, bye. Yeah. Steps of DNA replication. So you have two strands, two strands of replication along each replication fork. If you're taking bio, you should know this. You have a leading strand where the primase, which adds a section of RNA nucleotides to the onto the DNA, will lay out a primer, which is like a starting signal for DNA polymerase, specifically DNA polymerase 3, and adds nucleotides to the growing 3' end of the primer. So on the leading strand, the polymerase can just and go all the way down without stopping, because the new end, the exposed end here, is the 3' end. Does that make sense? The other side, the exposed strand, if you see how the replication fork is moving to the left, the leading strand is also moving to the left, but the lagging strand is moving to the right. Why is that? Well, that is because DNA is antiparallel, which means that if you were to just think of it as if you were to synthesize DNA this way, starting from here to the left, then the exposed end would be a 5', the 5' end of the nucleotide. So remember this, the polymerases, RNA polymerases, DNA polymerases, they can only add things to the 3' end of a nucleotide. So because of that, you need primers need to be continuously placed, and the polymerase has to go in the opposite direction, forming fragments called Okazaki fragments, joined. And then later, these different fragments are like stuck together by ligase. Yeah. Proofreading or repair. Enzymes can remove and replace incorrect nucleotides in mismatch repair. So DNA polymerases have an inherent mismatch repair system built, like once they lay down a nucleotide, they can proofread for whether or not it's like the correct match. If it's not, it'll remove it and try again. Nucleases can cut out damaged DNA and replace nucleotides by using undamaged strand as template. So in this case, this is called excision repair, where, for example, if you have a piece of DNA where one end is like kind of twisted, convoluted, and the other end is perfectly straight, a nuclease will come in and cut out the section of DNA that's like warped. There are many reasons for why this can be warped. UV rays causing thymine dimers is a big one. Do we have thymine dimers on here? No, we do not. Causing thymine dimers is a big one. Cut out damaged DNA, replace nucleotides using undamaged strands as template. So after they cut out that section of DNA, polymerase comes in and uses a straight strand, the one that's like undamaged, and and replaces and uses nucleotide pairing to rebuild the DNA. I have one more slide after this slide, so we might have to go like 2 minutes over. Yeah, that's all. One more important section of DNA these are called telomeres. So if we go back to the lagging strand, I don't think we have the time to explain why we need telomeres exactly, but just know that because of lagging strands, at the end of every piece of DNA, like on the lagging strand side of DNA, every single time DNA replicates, you lose an Okazaki fragment at the end of the nucleotide, at the end of the DNA piece fragment. Strand, that's what I was looking for. At the end of the strand, you lose an Okazaki fragment, right? If that were part of a protein coding gene, every single time you replicate, you would lose a functional part of a gene, of a protein, which would really mess up biological systems because of how intricate they are, how perfect they need to be. So the solution to this are telomeres. These are non-coding regions of DNA. You should probably know the nucleotide sequence. Telomeres are repeating units of TTAGGG, and just a bunch of these, like thousands of these at the end, maybe not that, like maybe 1,000 of these at the ends of each DNA molecule. One of the functions like we just described was that telomeres will protect gene shortening. So it'll instead of the Okazaki fragments cutting out a specific piece of like coding exon DNA, the telomere will instead get cut off. And because the telomere is non-coding, it's just protective. It doesn't have a phenotypic effect on the cell. Another thing is because there's only lagging strands in one direction on each strand, the DNA fragments will have different lengths. They'll have different lengths on each side of the strand. So like if this is one side, this is the leading strand synthesis, this is the lagging strand. This uneven DNA strand will usually it will be like a big alert signal. There's DNA damage. Well, we have to commit apoptosis. But because the telomeres are associated with specific enzymes and proteins that prevent that from happening. So it protects DNA. Yeah. And they also might have a cancer protective function where a cancer cell can only divide so many times before the telomeres are depleted and it'll get rid of protein coding genes, and then the cell will die. Not die, but be dysfunctional. But cancer often has a lot of ways to get around this. Like for example, they have enzymes that make telomeres, and build them to the maximum length at the ends generally in their cells. Did the last part make sense? Or just in general? All right, I'm not seeing any questions. And finally, chromosomes. The actual structure of DNA is not just a soup of double-stranded like DNA floating around in the nucleus. No, they're actually bound by proteins. These proteins are called histones, and 8 histone proteins form a nucleosome when they're wrapped around DNA. One fun fact about histones is that they generally have the amino acids arginine and lysine because because these are positively charged amino acids. DNA is negatively charged, so by containing positive charges, it'll more easily attract the DNA and bind to it. Nucleosomes are equivalent to fibers. domains chromosomes. Each chromosome has their own like little area. It's not like a random soup. It's actually quite structured. Each chromosome has its own space in the nucleus. And chromatin, the structure of chromatin can also affect DNA transcription. This is like a major way that we can control the protein products of our cells. Heterochromatin is compacted chromatin. If you methylate DNA, so if you add methyl groups to DNA nucleotides or to the histones, that'll make the DNA more compact and result in less transcription because the RNA polymerases can't come in and find the promoter. It's hard for transcription factors to come in too. There's just not enough space. However, euchromatin, uncondensed chromatin is euchromatin. So you acetylate, like you add like negatively charged groups onto the tails of the histones, and that pushes the histones apart, which results in more space for attachment of transcription factors and the transcription unit, like polymerases. You should know these two. Yeah, that's all I have for today. Thank you guys for staying over. Sorry for going over, but I hope that was helpful. And there were no practice problems at the end.